But I would like to highlight the fact that the distinctive feature of this family of pathogens is, as Brad mentioned, the locus of enterocytophagy, or which encodes all the genes that are involved in generating this fascinating macromolecular structure, which is the type exclusion system, along with translocators and effector proteins and chaperones and regulatory proteins, which is going to be the subject of the talk. And I'm going to be focusing mainly on EPIC, but also Citrobacter, a typical EPIC, which also have, or EPIC, which also have the lead, basically share also the regulatory mechanisms that I'm going to be talking about. So one of the important aspects when we actually study the regulation of pathogens, in this case, is that they were acquired by horizontal gene transfer. And that imposes a need for the bacteria to find a way to control the expression of all the new information that is acquired. And in order to do that, the new genes are not only regulated by a cascade and a myriad of different regulatory proteins that actually are persistent in the cell that is receiving or acquiring these new genes, but it's also controlled by genes that specifically are brought together in the pathogenicity island or in accessory genes that are somewhere else in the genome of these bacteria. And in the case of the lead, everything fits on the regulation of a very important gene that is called for locus, for lead encoded regulator, which is encoded in the first gene, and I'm going to show that, of the Li1 oper. And here I'm just showing the three groups of regulatory proteins that are involved in the regulation of this gene. So I'm not pretending that you're going to be reading in detail this figure. It's just a way to show you what is known so far about all the pre-existing or ancestral regulatory genes or regulatory proteins that are involved in controlling the expression, as I mentioned, of LER. So all these myriad, all this repertoire of regulatory proteins that were already in E. coli actually feed on the control of the modulation of that particular gene. Why is this important? And this is because when enteric pathogens travel along the intestinal tract, they are going to face different signals, different environmental cues that actually are acting somehow as the address or the signaling that is going to actually tell the bacteria where to colonize. So imagine all this repertoire of regulatory proteins acting as the GPS for the bacteria, because what all these proteins are going to be doing is helping the bacteria to reach a consensus response that is actually going to tell the bacteria this is your preferred niche where to colonize. So the bunch of regulatory proteins I showed in the previous slide are basically detecting all these signals. So the intestinal tract is a gradient of many different environmental cues, such as carbon sources, hormones, pH, oxygen, bicarbonate, etc., that are being detected by all these regulatory proteins. But the problem with these cues is that some of them might be telling the bacteria not to express the lead, while others might be telling the bacteria to express it. So the bacteria is dealing with all these cues to reach a consensus response that is going to basically tell the bacteria this is your preferred niche, and that's where you have to start the colonization process. So fortunately, in the lab, we can study the repression or the expression of the lead or the induction of the lead in two lab media. One is rich medium, LD, the urea growth, where actually E. coli grows happily. That's a condition where the lead is not needed, and that's what we think it happens in nature, where the lead is not required. It shouldn't be expressed because the energetic cost to express the type exclusion system is very high. And fortunately for us, a medium that actually induces the expression of the lead is tissue culture medium, the medium that we use to grow cells. And as Brett was showing, the interaction of EPEG with tissue culture cells helps to understand part of the mechanism that EPEG uses to infect a host. So this medium actually induces the expression of the lead, which somehow makes sense, although in other pathogens is not an inducing condition. So what we knew over time, and this fascinating journey that Brett was describing before, to understand how these pathogens actually work, what we knew is that basically, the basic state of the expression of the lead is to be off. And for the cell, for the bacteria to do that, they use a global regulator that is called HNS, for histone-like nuclear destructuring protein, which basic function is to structure the bacterial nucleus, but by doing so, it also represses many genes. And it's being called actually the evolution sentinel, because it basically helps the bacteria to keep these genes off by binding to different sequences and forming nuclear repressor proteins that keep their RNA polymerase away. So, as I mentioned, the basic state will be to have the lead in the off situation, and this protein layer, which as I mentioned is called layer because it's a lead encoded regulator, is a parallel of HNS. So it has similarity, it shares some homology with HNS. And basically what it does is to act as an antagonist of HNS. So when LAIR is expressed, it specifically binds to the regulatory regions of the lead, basically the promoters of all the lead operons, and displaces HNS. And in that way, it's acting as the master positive regulator of the lead. But interestingly, as LAIR comes together with the pathogen is the island, there is also a bicistronic operon that encodes two other regulatory proteins that we call GERR and GERLA for global regulator of the lead repressor and activator. And which is interesting by these two proteins is that they are co-expressed. And it's known that they actually interact to form heterotrimers. GERLA is a dimer, GERLA is a monomer, and basically what GERLA does is to interact with the HTH domain, the DNA binding domain of GERLA, to prevent GERLA from binding to the regulatory region of LAIR, where GERLA actually starts a regulatory loop, a feed-forward loop, that basically increases the expression of LAIR until it reaches a threshold that can compete with HNS on the lead operons. But along as GERLA, as I mentioned, GERLA is also expressed. And what we thought at some point is that the growth conditions, the difference they make, is that when we grow under repressive conditions, the heterotrimer is formed, and in that way, we keep the expression of the lead off. Under inducing condition, the hypothesis was that the heterotrimer will not form, GERLA is free, and then GERLA can bind to the regulatory region and then activate the system, the regulatory loop, and then the whole expression of the lead. So that was an easy way to explain how the system works. And if I have the time, what I'm going to try to show is what we learned more recently that actually brings us new ideas of how the system is working that is not necessarily as easy as we thought. So, how does HNS actually represses and how LAIR actually activates? I'm just going to quickly show you. This is a protein DNA interaction assay where we can see the interaction of either HNS or LAIR with three fragments that divide the regulatory region of this divergent regulatory region that controls the expression of two of the lead operons. So, as you can see here, the three fragments contain either these sequences that we call silencers, or it's just acting as a control. It's a sequence that is downstream of this divergent regulatory region. And as you can see here, as we increase the concentration of HNS, HNS first binds to the silencer 2, and then later binds to silencer 1, which explains how HNS might be forming this nuclear repressor complex to keep these promoters in the off situation. If you try the same experiment with LER only binds to this fragment. And that's enough to disrupt the nuclear repressor complex. It opens up, and then the RNA polymerase can activate both promoters and get expression of the two operands. So you can basically see the same result by removing, using transcriptional fusion, where you can actually follow the force of the promoters. And if you look at the transcription of the B2 promoter, basically what you see, you have a fragment that removes either one of the silencers or the other, you get expression that is LER independent. So when you have the full regulatory sequence, you only see expression in the wild-type apex strain, but not in the LER mutant or in a E. coli laboratory strain that doesn't have the B. So basically, it fits with that result that HNS is binding to these flanking sequences to basically repress all promoters. So working with HNS mutants is tricky because it has a pleiotropic effect. As I say, it acts as, it compacts the bacterial nucleus, but also represses many genes. You knock it out, and you get overexpression of many genes, and the mutant is basically sick. So a way to go around that problem, you can use a dominant negative version of HNS that it's able to form HNS dimers, but that particular monomer doesn't bind to DNA. And a dimer that has a one-type monomer and the mutant monomer cannot actually bind to DNA. So if you induce the expression of this dominant negative version at some point during the culture, you basically can release, and I just want you to pay attention on the ones that have the green bar. In the LER mutant, when you induce this dominant negative version of HNS, basically you get induction of the need. What we are seeing here is a protein secretion profile. Basically, we can recover secreted proteins from the supernatant and look at them in a Kumasi gel, SDS gel. And basically what you see here is a double band with the SPVD, which are the translocated proteins that were mentioned, and the ESP8, which forms the filament, and it's a way to follow that actually the D is being expressed. You can also do it by doing Western blot, using antibodies against proteins that are encoded within the D. So basically, when you express a dominant negative, you see induction either in the LER mutant or in a double mutant LER-GERLA, which is the other positive regulator. So that was a way to show that in the absence of HNS, LER is dispensable, which also tells us that LER is not a bona fide activator that recruits the RNA polymerase. It's basically an HNS antagonist. So this is the model of how we thought the system was working. HNS forms these nuclear repressor complexes. That actually is very interesting because these complexes can actually trap the RNA polymerase, but keep that transcription from going on. And that actually makes the system very efficient because when LER is present, the nuclear repressor complex is dissolved, the RNA polymerase is ready to go to actually activate those promoters. And that's why the response under inducing conditions is very fast. So how the system goes back to normal, and the reason why it happens is because by another mechanism, LER is repressed when the bacterium is reaching stationary phase, or the culture is reaching stationary phase. And at that point, LER is degraded. But HNS is present along the way. And when LER is degraded, HNS takes over again, its binding sites for the nuclear repressor complexes, and then turns the lead expression off again. I was talking about GERLA. So GERLA is this protein that actually directly activates layer expression. It's also encoded within the E. So the pathogenicity island also comes with its own set of regulators. GERLA is the activator of layer. And I'm going to be talking about another interesting characteristic that IPEC has. IPEC has a high molecular plasmid that encodes all the forming pilus, which is a very important pili that helps IPEC to bind to glial cells. But it also encodes another regulator that not only activates the BPO, but contains a gene that actually also activates the layer promoter in a very specific way under different growth conditions. So it has a way to actually coordinate the expression of plasmid encoded virulence genes with the lead pathogenicity. So that's another interesting story. How GERLA acts? Several years ago now, it was known, or we learned, that actually GERLA binds to a specific sequence that is between the -10 and the -35 promoter sequences, which is not normal for an activator. Usually activators either bind -10, -35 in the promoter to recruit the RNA polymerase, or somewhere else to eliminate a repressor. So it actually looks more like a repressor. However, and I'm not going to have the time to go into the details, but basically what GERLA is doing, and it was proposed several years ago, that based on the characteristics of the promoters that are target for layer, which are actually very particular because they don't have the canonic distance between the -35 and the -10 promoter sequences. It has an extra base, and it was proposed in the past that actually GERLA, by binding to this site, was correcting this difference in the distance between these two boxes that are very important for RNA polymerase binding. But the other characteristic that is important in these promoters is that the -10, which is very important to open the transcription complex, has Cs or Gs, which are not common in the -10 sequence, which is basically AT rich, because that facilitates the opening of the transcription bubble. So these changes are very important. And what we think now, and I don't have the time to go into the details, is that GERLA is not only binding here to be specific for the activation of its target promoters, but the binding in this place is because at some point it acts as a transient subunit of the RNA polymerase to help the RNA polymerase open up the transcription bubble in these promoters that are not canonic. Because basically, if you change the C's for T or A's, this promoter becomes GERLA independent. So this hypothesis that it was actually fixing the distance by making a structural change is not that right. And we think that the mechanism is a little more complex, and what we are trying to prove now is that actually GERLA actually interacts with one of the RNA's polymerase subunits. You're going to be seeing pictures of the people that actually did the work. So another evidence that we had at some point that actually started to change our mind is that, as I mentioned, the role that we have for GERLA is that it specifically binds to GERLA to prevent GERLA from binding to the layer promoter. However, based on the knowledge we have and knowing that in the absence of HNS, layer is no longer required, Christina did a simple experiment that we thought was going to work straightforward. Because when you overexpress GERLA in the wild-type strain, basically you see repression. So what we thought is that it titers GERLA. And that was easy, an easy way to explain the phenomenon. But what Christina thought is that by overexpressing GERLA in an HNS mutant, we were not going to see the same phenomenon. But bingo, it actually also represses. And that took Christina through a journey to actually learn that Gerlar is repressing the lead also in an independent way, not interacting with Gerlay, but perhaps interacting directly with DNA. So we still don't know if that's the case, but there is some evidence that points out into that direction. In fact, at the time, I'll show you additional data from that regard. So, but then one of the questions that we had at that point is what's the role of the heterotrimer, this interaction between the two proteins, if it wasn't only that it was a way for Gerlar to inactivate it. So I'm not going to go through all the details. I'm just going to want to show you that we have several Gerlar mutants. Two of them have an interesting phenotype because both of them have mutations in the DNA binding domain, so they are not able to activate the Ler promoter. But interestingly, one of them is no longer able to interact with Gerlar, but the other is. And this was important to use them as tools to show that by co-expressing wild-type Gerlar with a mutant that actually doesn't longer interact with Gerlar, Gerlar was still able to repress the Lee expression, but not the one that doesn't activate the Lee, the Ler gene, but still interacts with Gerlar. By co-expressing them, now Gerlay was being able to prevent Gerlar repression. So that brought us the idea that actually Gerlay not only has a positive regulatory role by activating Ler, but that might also have an additional role by preventing Gerlar from repressing the lead in this independent Gerlay interaction independent way to repress the lead. And we'll just keep that. Well, this is just going to show you that something that's interesting. As I mentioned, if we knock out HNS, the lead is de-repressed. But we also knock out Gerlar independently, you see high expression of the lead. So both are repressors of the system. One was a persistent regulatory protein, the other came together with the lead pathogen STI. Both of them, by knocking them out, with both of them you get this nice high expression and low secretion of lead-encoded proteins. So this was important because there should be a way how these two proteins are acting together, and I'm going to come back at the end with this. So the other thing is that the model that we had at the beginning, as I mentioned, is that it was an easy way to explain how the system worked by just hypothesizing that in LB, enriched medium where the Lee is repressed, the heterotrimer is being formed, and then by forming the heterotrimer, you have a repression of the system, and in DMEM, probably the heterotrimer wasn't being formed, and then you get activation because Gerlay was free. So these experiments basically are Western blots, native polyacrylamide gels, or in SDS page, where you can see expression of all Gerlay or Gerlar flagged or tagged with the flag epitope. And in native gels, you can actually see the interaction of both proteins with all of them repressed. So when you have Gerlar tagged with flag on the DMM or LB condition, you can see actually the expression of the Gerlar monomer, the Gerlar dimer, and then the Gerl-A Gerlar heterotrimer being formed. And interestingly, when you only have Gerl-A tag, you see where Gerl-A is, and you only see Gerl-A in the heterotrimer. So that was normal, that's what we were expected to see, but only in LB. So the point of this result is that the heterotrimer is being formed under both conditions. So once again, what was the role of having the heterotrimer under both conditions, where the heterotrimer was supposed to repress gene transcription? So that was one of the questions that we still have to fully answered, hopefully in the future. So once again, I'm showing here the interaction with the GERL-A mutant that doesn't interact with GERL-A anymore, and the one that still interacts. And this is a way to show that even in both conditions, you can see the interaction of the mutant when it still interacts. In this case, you see the heterotrimer, and since you are expressing GERL-A, you drive the formation of the heterotrimer, and in this case, where there is no interaction between the two proteins, you only see the dimer and the monomer of GERL-A. So, trying to understand what was the role of the trimer, what was the dynamics of GERL-A and GERL-R in the cell, to understand what was going on and how was the system repressing or reducing the system. And I'm also not going to have time to go through all the details, but a very bright student, Anna Alba, has been using fluorescent proteins and different techniques to try to understand the dynamics of these two proteins in the cell. So the initial hypothesis is that actually GERL-R and GERL-A probably were going to be colocalizing and forming heterotrimers all the time in the cytoplasm. And basically what Alma has been found using FRED, BFC, and single molecule tracking is that, interestingly, during inducing conditions, GERL-R and GERL-A at the beginning of the culture are more towards the membrane, or are more associated apparently with the membrane. And as the culture grows, what happens on the inducing condition is that GERL-A migrates or relocates towards where the DNA seems to be. If she does the same experiment in a K12, E. coli K12 strain, she doesn't see this relocalization. So it seems to have to do with the presence of GERL-A binding sites on the chromosome. Interestingly, when she uses the model that we had at the beginning. But then we have some preliminary data showing that actually GERL-R can bind to the LAIR promoter. And as you see here, these are again these shift assays where we can actually see the formation of a complex with the regulatory region of LAIR. The control is just a sequence further upstream of the LAIR promoter, which doesn't bind, GERL-R doesn't bind, and then you see the complex as the concentration increases. Interestingly, if you perform the GERL-R complex with the LAIR promoter, and then start adding increasing concentrations of GERL-A, what you see is that GERL-A at some point displaces GERL-R, releases the DNA fragment, but then starts binding to the same fragment, which suggests that under repressive conditions, GERL-R is also helping to keep the lead expression off. As GERL-A increases, or free GERL-A, and we still don't understand exactly what's going on with the growth conditions, but as GERL-A increases, GERL-A not only displaces GERL-R, binds the LAIR promoter to activate expression. If we do the same experiment with the GERL-A mutant that doesn't longer interact with GERL-R, you don't see the same displacement, you only see the release of the DNA fragment because this mutant still interacts with GERL-R, but not with the LAIR promoter. So we believe that a way for GERL-R to also help repressing the system is by binding directly DNA. Which we don't understand is that by looking at the structure of the sequence, GERL-R doesn't have features of a DNA binding. So we still don't know how it does it. So I'm not going to go through the details, but basically that simple scheme that I showed you before has got more complex because the lead has these two levels of negative regulation where GERL-R and GERL-A and GERL-R are playing dual roles in the regulation of the lead, as well as other targets that are in the chromosome. So these are the conclusions. These are the questions that we are still addressing. That I showed you some evidence. But then, just to finish, we are also addressing another question. All we have learned is from prototype strains, the strains that we use in all the labs. But what happens if you start looking at clinical isolates? There is a whole diversity out there. And Carmen Contreras, some years ago, just came out, started addressing the question if the regulation acts in the same way as in the prototype strain. And just to show you an example, because she found all sort of different phenotypes when looking at oppressive and inducing conditions. So what you see here is one of the clinical isolates that actually overexpresses the lead under any condition that she grew the strain on. And most interestingly is that when looking at the prototype strain, if you mutate GERL-A and PERC, this PERC is the other gene that also is able to activate the alert promoter, for the prototype strain you don't see the expression. So this is the double mutant. But when you do the same thing in this clinical isolate, the strain is still expressing the lead. This doesn't have to do with the fact that it has a defective GERL-R. The GERL-R caused by this strain is still active. Doesn't have to do with changes in the DNA sequence that might render constitutive promoters. It has to do with the presence of another regulator that we are trying to chase but not able to find so far. But basically what this is telling us, and there are some subtleties, even when comparing IPEC and IPEC or Citrobacter in the way they infect the cells, or how TIR acts, for example, is that the evolution has been able to generate different mechanisms for this family of pathogens to be able to infect a host. We still don't know if there is a correlation with the clinical symptoms that the child from where this strain was isolated, but one of the ideas is to probably later on understand if this has to do with the severity of the disease. So thanks to the lab and the actual actors that do the work, and collaboration that we have, long-term collaboration with Brett and some other groups that have helped us to understand the regulation of the E in this activity. Thank you.